How Antibody Therapy Treats Breast Cancer by Laila Rabaa. Why do this topic? Every year, around 42,260 people die from breast cancer. It is a horrible disease that completely changes people's lives, and I wanted to learn about one of the groundbreaking treatments that have saved many people. And luckily, we are starting to gain more than one approach to treating this horrible disease. Antigens. They are proteins that are found on the surface of a pathogen, and each antigen is unique to that pathogen. Antibodies are what helps keep our immune system healthy. They are made up of 4 separate chains, 2 light and 2 heavy chains, to form a Y. They are proteins that look for a specific antigen to attach to. T cells' main job is to protect our body from foreign invaders. Once it has encountered a foreign antigen, they are activated and create antibodies which are told to fight that antigen. Antibodies then recruit T cells, a type of white blood cell which then proceeds in assisting in destroying the pathogen or cell that has that specific antigen by either directly killing the antigen target and or releasing chemicals known as cytokines to signal other cells to kill the target antigen. White blood cells are critical to a healthy immune system, though they only account for 1% of our blood. They are important to us as humans, and without them, our body would not function properly. There are many types of white blood cells, but the one used in this process is lymphocytes, which create B cells and T cells. Monoclonal antibodies to treat disease was a huge milestone achieved in 1984. In order to produce large quantities of a specific antibody to target a specific antigen, production methods were developed, and now scientists are able to manufacture monoclonal antibodies at a very large scale. They are identical antibodies produced from a single B cell clone. Monoclonal antibodies are created by using mice and injecting them with the target antigen. This activates the B cells to create antibodies to fight that specific antigen. Scientists then remove the B cells and harvest them from the mouse's spleen. Once the B cells have been harvested from the mouse's spleen, they end up fusing together with tumor cells. This occurs because B cells don't divide, but tumor cells do. This is known as a hybridoma cell. The hybridoma cells reproduce quickly and can make clone cells. They are then combined with an anti-cancer drug. They then match perfectly with the specific antigen. Breast cancer. Breast cancer information. Every year in the U.S. alone, there are 266,000 new diagnoses of breast cancer. A woman has a risk of a 1 in 8 chance of developing breast cancer during her lifetime. Scientists say that around 5 to 10% of breast cancers are linked to gene mutations passed through generations and families. Breast cancer stages. There are 5 stages in breast cancer. Stage 0, the patient has a 100% chance of survival. Stage 1, a patient has a 100% chance of survival. Stage 2, a patient has a 93% chance of survival. Stage 3, a patient has a 72% chance of survival. and stage 4, a patient has a 22% chance of survival. HER2 is a gene mutation that is passed down and can result in positive HER2 breast cancer. Normally, HER2 is a breast cancer receptor which controls how healthy breasts repair, grow, and divide themselves. Though, 25% of the time, it does not work that way. When HER2 makes too many copies of itself, it makes the breast cells divide and grow in an uncontrolled manner. Breast cancer cells with HER2 gene amplification of HER2 protein overexpression are called HER2 positive. On the left is an example of a normal HER2 breast cancer receptor, while on the right is an example of a HER2 positive breast cancer receptor where the cells have multiplied in an uncontrolled manner and there are too many receptors. Herceptin, also known as trastuzumab, is an immune-targeted therapy that helps fight breast cancer. It attaches itself to HER2 receptors in the breast cancer surface cells to stop them from receiving growth signals. Herceptin not only inhibits HER2 signaling pathways, but also initiates immune-mediated responses against HER2 overexpressing cells. Herceptin then attaches itself to HER2 receptors and stops the breast cancer surface cells from getting signals. There are many benefits to taking this drug. It treats HER2 breast cancer, and 40% were less likely to die from cancer. As well, it lowers the reoccurrence of 36% less likely to have cancer return. Though there are benefits, there are also risks to taking this drug, such as high blood pressure, headaches, hot flashes, nausea, and body pain. Thank you for watching. I hope you enjoyed this video.